Okay, everyone, we're going to start talking about respiration now. We spent the last lecture discussing the cardiovascular system and how blood, the respiratory medium, is circulated throughout the body. And now we're going to talk about how that circulatory medium, blood, and the cells and its constituents it contains are actually used to accomplish gas exchange. So, of course, the purpose of the respiratory system is the exchange primarily of oxygen and CO2, but also to a lesser extent ammonia or NH3, between the environment and the tissues which either produce or consume those molecules. The exchange can be broken down into essentially the 5 basic steps that are described here. So the first one is the transport of water to and across the respiratory surfaces, or the gill. So just how the water is moved across the gills to facilitate gas exchange. The second step is the actual exchange of these gases or molecules at the respiratory surface. So this is the physical uptake of oxygen and the release of carbon dioxide and ammonia. The third step is transport of those molecules from the respiratory surfaces to the tissues that need them. So transport of oxygen to the cells and pickup of CO2 and ammonia. The fourth step is exchange of the gases at the tissue. So again, this is the drop-off and pickup exchange that occurs at the capillaries. And then the fifth step is circulation. So this is, again, moving the carrier medium from one site to another. In this particular context, it's easiest to think about this fifth step as being the transport of the deoxygenated blood that is now carrying additional carbon dioxide and ammonia back to the gill to be renewed, reoxygenated, etc. Now, of course, fish must have a very efficient respiratory system to compensate for how difficult gas exchange is underwater. And of course, when I say that something is difficult, a process is difficult, in the context of this course, that means energetically expensive. So fish have to be efficient to compensate for how energetically costly it is for them to acquire oxygen. So if you consider that atmospheric air, what we breathe in and out, has roughly 20% oxygen in it, we compare that to water. Depending upon temperature, atmospheric pressure, salinity, and other factors, it will have less than 1% oxygen. So it has substantially less oxygen in it. Now, in order to gain access to the same amount of oxygen that would be found in atmospheric air, the fish are going to have to move 30 times as much of the respiratory medium, water in their case, to gain the same amount of oxygen. So that either means that they have to expend 30 times more energy to get the same amount of oxygen, or their lung or their respiratory structure, gill, what have you, would have to be 30 times as large or 30 times as efficient. Another thing to consider here is that when we talk about moving a respiratory medium, air in the case of humans, water in the case of fish, there is a certain amount of energy that's associated with just physically moving that medium. So it's fairly easy for us to inhale and exhale air because it's a gas. But that first step that we talked about in the previous slide, the transport of water to and across the gills of fish, is not energetically easy. It's not energetically inexpensive. And this is because, of course, water is a liquid and it's much more dense and heavier and more difficult to move than air is as a respiratory medium. Now, depending upon the environmental conditions and the composition of the water, this situation may be even more dire with respect to availability of oxygen and the complexities of acquiring it from the environment. So, a couple of things to note here on this table. As water temperature increases, the solubility of gases decrease. Now, this is true for all gases that may be dissolved in water, and that includes oxygen, of course. There is actually the same relationship with salinity. As salinity in the water goes up, the solubility of gases goes down. So the way that I kind of think of this is there's only so much room in the water for dissolving stuff, whether that stuff is gases or salts. So if there's salt already dissolved in the water, it's going to push out or take up some of the room that would have otherwise been occupied by gases, including oxygens. So what you can see here is the relationship between increasing temperature and the maximum saturation of oxygen in water in either milligrams per liter or as a partial pressure of oxygen. So of course you can see that relationship, that as temperature increases, you see a pretty striking decrease in the maximum solubility. Of course, this is the maximum. This does not mean that just because your water temperature is 15 degrees that your oxygen availability is 9.8 milligrams per liter. More often than not, if you're talking about natural systems, the oxygen availability is substantially lower than that. Now, in the case of artificial systems in fish hatcheries, research systems, etc., it is quite common for the gas to be supersaturated. So at 15 degrees in a fish hatchery, you might actually have more than 9.8 milligrams per liter in the water. But again, that's a pretty narrow set of circumstances, and it's artificially induced in those environments. So again, efficiency is really key. Fish have to be efficient in terms of acquiring oxygen. We just discussed that the fish have to move 30 times more media for the same amount of oxygen as would be available in atmospheric air. And again, understanding that water is 800 times more dense than air, fish are going to have to move about 24,000 times more weight in respiratory medium, in water, to get the same amount of oxygen at their respiratory structure, at their gills, relative to what we have to do to get air across our respiratory structures or our lungs. Now, there are a number of different structures that may be used for gas exchange. Of course, the majority of this lecture is going to be focused on the gill because that's the primary respiratory structure for most fishes, but there are other alternatives. The so-called air-breathing fishes utilize what is essentially a rudimentary lung, or a quote-unquote lung. In this case, these fish are adapted to use a physostomous swim bladder as the site of gas exchange. So these fish, when they're at the surface, they can gulp air, force it into their physostomous gas bladder, because again, in physostomous swim bladders or gas bladders, that connection between the gas bladder and the esophagus is maintained throughout through metamorphosis and adulthood in fish. So again, once the respiratory medium, the air is in the gas bladder, there can be gas exchange there through the tissues that form the inside of the gas bladder itself. Now, this can either be facultative, in other words, it provides supplemental respiration, or it can be obligate in the case where this is the primary means of respiration in these fish. So you can see gouramis, for example, are obligate air breathers. So these fish don't have extremely functional gills, and so if they are unable to access the surface of the water, if they're not able to reach the atmosphere and gulp air, essentially over time they would ultimately drown, so to speak, because their gills are largely inadequate to supply the oxygen that they need. So they require the use of their swim bladders, their rudimentary lungs, to supply oxygen. So they are considered obligate. Now, in the case of plecostomus or bettas, these are considered facultative, so they can get by utilizing their gills as a primary site of respiration. But under circumstances where oxygen may be limiting in the water, they can certainly utilize their swim bladder to supplement that and provide some additional respiratory function. Now, dependence upon air breathing has actually rendered some species virtually amphibious over evolutionary time. So you can see a couple of these species here, like the walking catfish or mud skippers, which in addition to being quite ugly, are also rather unique in the sense that they are obligate air breathers and actually are very capable of utilizing a terrestrial environment. So in the case of the walking catfish, if conditions in the water body that they're in are unfavorable, for example, there isn't enough food or the ammonia concentrations get very high, they literally can just get up and walk out of the pond and find something new. It's the same thing with the mud skippers. So this is kind of an extreme conclusion to obligate air breathing and utilizing this rudimentary lung. Now, the skin is also another potential site of respiratory gas exchange, so cutaneous gas exchange, and it's most common in fish larvae. Like we talked about in the circulation and cardiovascular system lecture, the presence of a closed circulatory or cardiovascular system is a real advantage for having larger body sizes because you can connect portions of the body that would otherwise be very distant from the external environment to the external environment via that bloodstream. So by maintaining that connection, you can facilitate exchange of materials, including oxygen and carbon dioxide. But in the case of very small individuals, including larval fishes, most of the tissues in the body aren't all that distant from the external environment, and they have a very high surface area to body volume ratio, and so gas exchange can be a great supplement to these larval fishes as their cardiovascular system continues to develop and their gills to continue to develop. Now, of course, most fishes will grow out of this cutaneous gas exchange. As their body size increases, they will become more greatly reliant on the gills as respiratory surfaces, or in the case of air-breathing fishes, the modified physostomous gas bladder as the primary site of gas exchange. But of course, gills are the primary site of respiration for most fishes, and so that's where we're going to spend most of our time discussing. So the basic structure consists of the gill arches, the primary lamellae, and the secondary lamellae, and you can see these here. So the gill arch is providing largely structural support, so there's no gas exchange there. You can see the primary lamellae on the right-hand side as the kind of major fronds, so to speak, of the gill. And then the secondary lamellae, this is where the actual gas exchange occurs, are essentially a series of little flaps of skin that extend along the entire length of the primary lamellae. In terms of completing that first step of movement of water across the respiratory structures, you can see that illustrated here. So this is essentially a cross-section through the head of the fish. So you can see the mouth there on the left-hand side, so the water is flowing in through the mouth and flowing linearly across the primary and secondary lamellae back out through the back of the opercula. Now, the important thing about this is that the flow of water through those gill filaments is laminar, so it's a consistent flow, it's not changing direction, and it's parallel to these lamellae. That's key because if you have flow that is parallel to the lamellae, then the flow of the blood is actually counter to the direction of water flow. And as we'll talk about in a few slides, that counter-current exchange is a key element of establishing the efficiency of oxygen acquisition at the gill. And this is just a close-up micrograph of an electron scanning micrograph of what those gill lamellae look like. So you can see there are two primary lamellae here, and then lots and lots of the secondary lamellae, those little flaps along their length. There are a number of different cell types and tissue organization levels at the gill. That's illustrated here. You can see, first of all, there's the gill raker, number 1 there, shown on the left-hand side of the structure. The gill raker is covered with mucosal epithelium, so this is basically modified skin-like structure. Then number 3 there refers to the basement membrane. This is primarily structural, as is number 4, the submucosa, the bone, which is shown there, number 5. 6 includes a little bit of adipose tissue. Again, most of what's on the left-hand side here is, I don't want to say non-functional, but it's not directly involved in respiration. The respiratory elements are on the right-hand side of the figure here. So number 7 is showing the efferent branchial arterioles, and the number 8 is showing the afferent branchial arteries. So this is blood that is leaving and coming to the gill. And again, the direction of that flow is going to be very important, as we'll see. Of course, you can see number 9 and number 10 illustrating the primary and secondary lamellae. And I think what's important here is to appreciate that although on the previous slide the electron scanning micrograph made it appear as though the gill filaments were kind of large and chunky, all things are relative. These are very fine structures, and they need to be in order to facilitate that gas exchange. Okay. So if we zoom in a little bit closer, on that same slide, you can see again how thin and delicate these structures are. The basic structure of the primary lamellae is basically two single sheets of epithelial cells that are held apart by so-called pillar cells. So you can see number 5 is referencing the pillar cells. These are basically just kind of what they sound like. They are pillars that keep the two very thin single cell thickness layers of epithelium separated. Otherwise, these two layers would likely collapse together and they would prevent red blood cells from moving through those secondary lamellae. So you can see lots and lots of red blood cells in, for example, number 9, the central venous sinus. So this is blood that's coming into the gill for reoxygenation. And you can see individual cells moving out into each of the secondary lamellae. It's basically just wide enough to accommodate one layer of red blood cells. So again, we're talking about very, very thin, very, very delicate structures. It's also important to note that there are a number of other different types of cells that are listed here. Epithelial cells, mucosal cells, the pillar cells we've already talked about, the chloride cells as well, which I don't believe are shown on this slide. They may be on the next slide. The gill is responsible for a lot more than just gas exchange, and we'll talk about that in the next lecture when we discuss osmoregulation. But for our purposes here today, the primary cells that we need concern ourselves with are those erythrocytes or red blood cells and the epithelial cells of the secondary lamellae. Again, zooming in here a little bit more closely, yes, you can see in this case a few more of the different types of cells. So you can see once again the erythrocytes, the red blood cells, highlighted for example at number 10. You can see that red blood cell is essentially occupying the entire space available within the secondary lamellae. You can also see the pillar cells, which are providing that structural support to keep those flaps from kind of collapsing down upon themselves. And you can also see some of these additional cells that I mentioned, like the mucus cells shown number 6, or the very large chloride cells. The mucus cells, of course, are producing mucus to keep the surface of the gill lubricated as well as free of bacteria. So of course these very delicate structures, because of their delicacy, they are vulnerable to bacterial attack or attack from other types of protozoan parasites, etc. And so part of the function of mucus is to slough off any of that bacteria or protozoa that might otherwise be trying to attack the gill. So the mucus cells, although all they do is produce mucus, are very key to maintaining the functionality of the gill. The chloride cells we will discuss in greater detail, but these are the primary means by which fish osmoregulate and maintain proper levels of salts inside their body relative to the environment. And again, just another a scanning electron micrograph to show you what this looks like in a little bit more real-life type of image. So you can see each of those secondary lamellae have been sliced in half, and you can see many of the pillar cells there, which again are providing that structural support so that the red blood cells can move freely through the spaces between and acquire oxygen and deliver CO2 and release that to the environment. Another image, another view of what these things look like. So you can see in the upper portion there is the secondary lamellae. So you can see the structure of it, the flap structure itself, and you can also see in the arrow the direction of flow. So the water is going to be flowing in one direction, the blood's flowing in another direction. Again, that countercurrent exchange being critical to ensuring maximum efficiency of oxygen acquisition. Okay, another cute little picture here showing the direction of water flow through the gills of the fish. You already saw this in a slightly different diagram earlier in the lecture. This is also showing a little bit greater detail regarding the flow of the blood. So they're showing deoxygenated blood in blue, oxygenated blood in red, and they're also highlighting the afferent and efferent vessels. So afferent vessels, the way that I keep it straight in my mind is I think that the A at the beginning of afferent stands for arriving. So this is the blood that is arriving at the gill. Those are the afferent structures. Of course, they are blue because the blood that is arriving at the gill has yet to be reoxygenated. Now, of course, as the blood flows through those capillary beds in the secondary lamellae and then exits the gill, those vessels are considered efferent. And again, the way that I keep this straight in my mind is I think that the E at the beginning of efferent refers to exiting. So we have blood that's exiting the gill, and of course, when it's exiting the gill, it has been reoxygenated and is depicted by the red color here. So you can see here the water flow in those dark blue arrows at the bottom of the screen going opposite to the flow of the blood through those secondary lamellae. So again, the countercurrent exchange, the water flowing in one direction, the blood flowing the opposite direction, ensures maximum efficiency by maintaining a concentration or a tension gradient regardless of the actual saturation levels of oxygen in the water versus the blood. So if you look at the bottom figure, actually, this would be showing concurrent flow. This isn't what happens in fish, but you'll see why here in just a moment. You can see that the water is flowing in one direction and oxygen is moving from an area of higher concentration or higher tension pressure in the water, starting at 100% saturation in the water that's coming into the gill. It starts to diffuse into the lower concentration or tension in the blood, shown at the bottom of that figure. And so we have it basically averaging out to a 50-50 split between the water and the blood. That is the maximum enrichment that you can achieve in the blood because once it reaches 50-50 and it equilibrates between the water and the blood, there is no more concentration gradient, there is no tension gradient, and so there is no impetus to drive oxygen moving into the blood. So if you have a concurrent exchange system, this is the maximum concentration you could get, only about 50% of what would have been available in the water. Now, of course, with a countercurrent flow, you can achieve much greater enrichment of the blood, and that's what you see here at the top. So, of course, as the water comes in, initially it's carrying all of the oxygen that it could possibly carry, and so it's diffusing into the blood. But as it becomes depleted, it's getting down to maybe 40 to 15% of the total oxygen it was carrying when it entered the gill. Again, it's losing its oxygen because that's diffusing into the blood. But at that point where its oxygen capacity or oxygen carrying capacity is reduced, that is where the afferent blood, the deoxygenated blood, is coming in. So even though the oxygen availability in the water is reduced, it's still higher than the very low levels that are in the afferent arriving blood supply. So you can see the concentration gradient there is 15% to 5%, so it still favors an influx of oxygen into the blood. And of course, as that blood moves from the afferent vessels into the efferent vessels, it's being enriched with oxygen all the way. And even though it gets to the 90% enrichment level, relative to the water that it's interacting with that's just coming into the gill, there is still a concentration gradient there that facilitates diffusion of oxygen into the bloodstream. So you can achieve maybe 90 or better percent enrichment in the case of a countercurrent exchange system, much, much greater than what you can achieve with a concurrent flow exchange. Another perhaps simpler diagram showing the same sort of thing. These countercurrent exchangers maximize the transfer of oxygen as well as a variety of other different things between media that are found in different places in the body of the fish. So we're talking about it now in the context of oxygen exchange with the water, between the water and the blood. So you can see the concentrations, or rather the pressures of oxygen in the water versus the blood in millimeters of mercury here. So again, you can see the direction of the water moving one way, the blood moving the opposite, and the tension or pressure gradients continuously favor diffusion into the blood regardless of where you are along those vessels. So this is certainly beneficial in the case of oxygen acquisition. Keep in mind that it works the exact opposite way for carbon dioxide. So carbon dioxide would be at very high levels in the afferent bloodstream. Those would diffuse out into the water. As those concentrations in the blood decline, theoretically the concentrations in the water could be increasing, and so CO2 diffusion will continue to be facilitated by this countercurrent exchange as well. But again, countercurrent exchange or countercurrent systems don't exist solely in the gill. The gill is a major site of this. We already talked about the one that exists in the gas bladder to facilitate recapturing of the CO2 and lactic acid that is produced by the gas gland cells. But there are other countercurrent exchange systems throughout the body of the fish. One in particular that is important that we'll discuss later on is the countercurrent exchange of blood vessel to vessel in the eyes of many cold water fishes that is not to facilitate exchange of gases but is meant to recapture heat that is generated by the musculature. So for highly active species that are in cold water systems, their musculature blood is the respiratory medium, of course. It is the medium for circulation of nutrients and other products as well. But red blood cells are the primary drivers of gas transport in most fishes. Red blood cells are the most abundant cells in the blood of fish, and they represent typically 20 to 30% of what's called the packed volume. In other words, a hematocrit of 20 to 30% is pretty typical. So for those of you who haven't done hematocrit before, essentially what you do is you take a what's called a microcapillary tube. It's a small glass tube. That's what I'm illustrating here. You draw up a sample of the fish's blood in that tube, and then you centrifuge it to separate out the cells from the rest of the plasma. Or the liquid aqueous portion, the acellular portion of the blood. And so typically of the entire volume of blood that was collected after centrifugation, usually the packed volume or the cellular volume typically represents about 20 to 30%. Okay. Now again, the number of red blood cells or the absolute hematocrit, whether it's 20%, 30%, 50%, will depend upon a number of factors that we'll talk about in a moment. Now, unlike most other vertebrates, fish's red blood cells are nucleated. There's really no implications to that other than when you look at them under a microscope, they actually have nuclei as opposed to higher vertebrates. Most higher vertebrates do not have nucleated red blood cells. So what makes red blood cells so special and the primary driver of gas transport? It's the fact that they possess hemoglobin. Hemoglobin is a pigment, but it is a pigment that is specialized that allows for oxygen, CO2, and other molecules to be effectively transported. So again, the absolute concentration of hemoglobin within the red blood cell, as well as the number of red blood cells that are circulating at any time, will vary based on a number of different factors. So habits, the fish, so their activity levels will influence both of these things, hemoglobin concentrations and the number of red blood cells. Generally speaking, fish with higher activities tend to have more red blood cells and higher hemoglobin concentrations within them to facilitate that high level activity and ensure rapid oxygen acquisition and delivery. Habitat will also influence the numbers of red blood cells and hemoglobin concentrations. Generally speaking, the lower the dissolved oxygen availability in the environment, the more hemoglobin and red blood cells you will have to compensate for that and ensure that the limited amount of oxygen available in the environment is captured. Seasonal changes will also influence red blood cell numbers. So again, this is primarily driven by water temperatures. So there are fewer red blood cells needed in the wintertime, typically because dissolved oxygen is more commonly available at higher concentrations because of the greater solubility of oxygen at colder water temperatures. The other reason that red blood cell numbers tend to decline in the wintertime is because red blood cells contribute quite greatly to the viscosity of blood. And so as the viscosity of the blood increases as temperature declines, it's going to be much thicker, much more difficult to circulate. And so, as we discussed in the cardiovascular system lecture, to compensate for the challenge of circulating viscous blood in the wintertime, fish will usually respond by decreasing the number of red blood cells that are circulating. Swimming activity, we already discussed a bit in terms of overall activity levels. Generally speaking, the more active a fish is, the more red blood cells it will have. Now, pollution here is actually an interesting one in that you tend to have higher levels of red blood cells when you're dealing with nitrite toxicity, for example, or other types of water pollutants. This is, again, to compensate for the effects of those pollutants on gas exchange. Nitrite, for example, irreversibly binds hemoglobin and makes it unable to transport gases effectively. So if more of your hemoglobin and more of your red blood cells are inactivated by binding with nitrite, the fish usually respond by generating more red blood cells and more hemoglobin to ensure that gas exchange is still possible. So you may have a higher number of red blood cells during exposure to pollutants, or the turnover rate may be higher. So fish may be breaking down and producing more red blood cells to compensate for the effects of the pollutants on the functionality of the red blood cells themselves. So again, hemoglobin is this pigment, this unique molecule that is the primary adaptation that allows for efficient oxygen and carbon dioxide uptake and trafficking. Now, not all fish have hemoglobin, but those that do enjoy greater oxygen capacity within the blood. In fact, the presence of red blood cells and hemoglobin in the bloodstream can increase the oxygen carrying capacity of blood up to 20 times what would normally be soluble in the aqueous portion of blood itself. Remember that the solubility of gases, including oxygen, in water is going to be influenced by the presence of solutes. Now, the majority of the blood is water, but it's water that has a lot of other things dissolved in it, like salts and proteins. So the absolute solubility of gases in plasma, the liquid portion of blood, is pretty low. But by the virtue of hemoglobin and its ability to bind oxygen and allow the blood to carry it without dissolving it, you can increase those carrying capacities substantially, again up to 20 times what would otherwise be soluble just in the plasma portion. Hemoglobin has what's called a tetrameric structure. So hemoglobin is actually 4 molecules together. You can see there are 2 red molecules and 2 green molecules. These are the so-called beta chains and alpha chains that comprise a hemoglobin protein. You can see that there are actually 4 what are called heme groups. So each of these structures do slightly different things. So the alpha and beta globin chains, these will actually bind carbon dioxide and hydrogen ions. And the heme groups themselves, these will actually bind oxygen. The heme groups are what contain the iron that we typically associate with blood and with gas transport. So oxygen binds to the heme groups and the iron within them. Carbon dioxide and hydrogen ions bind to the alpha and beta chains. And it's the interaction between these various ligands, or things that bind to the molecule. So the interaction between oxygen, carbon dioxide, and hydrogen ions will affect how the molecule, how hemoglobin binds to other things. So as we'll discuss, if there's a lot of oxygen bound to the heme groups on hemoglobin, that means there's less room for carbon dioxide or hydrogen to bind to the alpha or beta chains. Conversely, if there's a lot of hydrogen and carbon dioxide bound to the alpha and beta chains, there's less room for oxygen to bind to the heme groups. And this might seem kind of simple and not very relevant, but it's those interactions, what are called allosteric interactions. In other words, the oxygen, carbon dioxide, and hydrogen all jostling for space on the hemoglobin molecule. It's those interactions that drive the ability of hemoglobin and the red Now, we talked about this a little bit already when we talked about gas deposition in the swim bladder, but hemoglobin and changes in the oxygen carrying capacity of blood are affected by three different interrelated effects. So first, there's the Bohr effect. Okay, the Bohr effect is a decreased affinity of hemoglobin, which I'm abbreviating here as HB, to oxygen. Okay, so essentially a reduction in blood pH or plasma pH decreases the binding affinity or the stickiness of hemoglobin to bind oxygen. Okay, the root effect is related, and it's essentially a decrease in the oxygen carrying capacity of blood. Because there is less oxygen bound to hemoglobin, because of the Bohr effect, more ends up being dissolved in the plasma itself, leading to a total reduction in carrying capacity. Recall that oxygen that's bound to hemoglobin, quote unquote, doesn't count. It doesn't contribute to the solubility of oxygen in the bloodstream, in the plasma, because it's not really dissolved in the plasma. It's bound to hemoglobin. So as long as the oxygen is bound to hemoglobin, you can bind and carry more and more and more in the bloodstream without being subject to the solubility of oxygen and gases in the plasma itself, which, as we've discussed, is quite low. So if the Bohr effect decreases that affinity, so you don't have the same stickiness of the hemoglobin, more of that oxygen will break away from the hemoglobin, and now it will become solubilized in the plasma, and it will contribute to the total solubility of gases, and you will be subject to the rules as far as salinity and temperature affecting gas solubility in aqueous phases. So the root effect is a function of the Bohr effect. Bohr effect is the decreased affinity or stickiness of hemoglobin for oxygen. Root effect is a consequence of that, in that it is a decreased carrying capacity of the blood for oxygen. Now, the third effect here, the Haldane effect, is essentially that deoxygenated blood or deoxygenated red blood cells have a greater affinity for CO2 and vice versa. So, in other words, when oxygen is dropped off at the cells that need it, the oxygen is becoming less attracted to the hemoglobin, therefore it is released and delivered to the cells. But at the same time that hemoglobin becomes less sticky towards oxygen, it becomes more sticky towards carbon dioxide, which is a good thing, because when the hemoglobin is decreasing its affinity for oxygen and releasing it to the tissues that need it, those tissues that need oxygen likely have high concentrations of CO2. The CO2 is now more attractive to the Bohr-affected hemoglobin, so the CO2 is attracted to the hemoglobin, it gets picked up, and then ultimately transported back to the gill for release to the environment. So again, all of these three things are interrelated and are largely the effect of decreasing pH or increasing hydrogen ion availability, affecting the stickiness of hemoglobin relative to oxygen and relative to CO2. So these are, this figure is a diagrammatic representation of the Bohr and root effects. This is also what's called an oxygen dissociation curve. So on the x-axis you have the partial pressure of oxygen in the blood, and then on the y-axis you have the percent saturation of hemoglobin. This is how much of the hemoglobin is bound with oxygen. Okay, so in the red line you have a typical curve at a pH of 8, let's say. That's pretty close to what's considered a typical circulating pH in the bloodstream of most fish. So as you increase the partial pressure of oxygen available, more of that oxygen becomes stuck to the hemoglobin. So the hemoglobin becomes completely saturated with oxygen as you approach approximately 80 millimeters of mercury pressure. If, however, there is a reduction in pH, so for example, this blood is delivered to white muscle cells that have produced a lot of lactic acid, and that lactic acid is then driving down the pH, maybe down to a more neutral pH actually, pH of 7, that effect is going to decrease both the affinity and the capacity. So you have the root effect, which is a decrease in the capacity. So the maximum saturation of hemoglobin you can get at that point at a pH of 7 is only 80%. And the Bohr effect is shown there by a shift of the diagram, shift of the curve to the right. So to achieve the same level of saturation, you have to have greater availability of oxygen in the blood. In other words, you have to get the same level of saturation, to get the same amount of oxygen to stick to hemoglobin, you really have to drive the pressure up and essentially force the oxygen to stick to the hemoglobin, even though it doesn't really want to. So this shift to the right is the Bohr effect. That's the loss of affinity. You have to work harder to get the hemoglobin to stick to the same number of oxygen molecules. And the shift down is the root effect. That's the reduction in total capacity, the total carrying capacity of the blood. Now, every fish is going to have its own oxygen dissociation curve, but the specific shape of that dissociation curve and how it responds to Bohr and root effects is going to be different depending upon that species, the environment it's adapted to, and its specific form of hemoglobin. So here you're looking at two oxygen dissociation curves for two very different species. We've got trout shown in green, which is a cold water adapted species, typically inhabiting environments that have very high oxygen availability. And then we have bullhead, which of course is a warmer water species that's adapted to living in environments that are typically limiting with respect to environmental oxygen. And so you can see the shape of the curves here is very different. The bullhead is adapted to those low oxygen environments, and it has a very high affinity hemoglobin, a very sticky hemoglobin. And so it achieves very high hemoglobin oxygen saturation, even at very low environmental oxygen availabilities. So along the x-axis here, even for low values of available oxygen in the environment, the saturation of the bullhead's hemoglobin is much higher, substantially higher than that of the trout. Now, of course, the trout is adapted to those high oxygen environments, so it requires much higher pressures, much higher availabilities of oxygen in order to achieve the same full saturation of hemoglobin. But again, keep in mind that this fish is inhabiting environments that are rich in oxygen. So the fact that they need higher pressures, higher availability of oxygen to achieve full hemoglobin saturation isn't really a problem, because those higher availabilities are typically available. Now, you could argue, well, wouldn't it be better for all species to have a bullhead type hemoglobin regardless of environmental dissolved oxygen availability? So even though the trout typically doesn't live in environments, typically isn't exposed to low oxygen availability in the environment, for those circumstances during which it was exposed to low oxygen availability, if it had bullhead hemoglobin, it would actually benefit from that. And that is true, but there's a disadvantage to that in the sense that the hemoglobin from the bullhead is very sticky and has a very high affinity for oxygen. That makes it advantageous in terms of oxygen acquisition from the environment, but it makes it somewhat disadvantageous in terms of delivering that oxygen to the tissues that need it. Remember that sticky hemoglobin is necessary for taking up oxygen at the gill, but that stickiness needs to be reversed by virtue of the Bohr effect to release the oxygen to the tissues that require it. So if all fish had bullhead-like hemoglobin, they might be better capable of coping with periodic reductions in oxygen availability in the environment, but they would be less able to deliver oxygen to the tissues in a rapid manner. So for a species that's fairly active like a trout, that really wouldn't be an effective approach. They need to be able to acquire oxygen and deliver it to those muscles that are working very actively during swimming behavior. Bullhead, as a more sluggish, bottom-feeding species, doesn't have the same metabolic demand of the tissues because it's not as active, and so the issues of releasing oxygen at the tissues are worth the trade-off for being able to have a greater ability to acquire limited oxygen from the environment. So we talked about the Bohr and root effect, and you can see that listed here for these different curves. So you can see both the loss of capacity, the downward movement of the curves, the suppression of the peaks of the curve, and you can also see the loss of affinity, that right-hand shift in the curves. The reason you see this shift, this root and bore effect occurring for these different curves, is as a function of the pressure of carbon dioxide in the bloodstream, excuse me, or at the tissues rather. So the orange curve represents the curve, the oxygen dissociation curve for a species, trout in this case, at zero availability of carbon dioxide. But as those carbon dioxide levels creep up into 3 and 8 millimeters of mercury pressures, you see the bore and root effect taking place. Again, keep in mind that this is because CO2 acts as an acid. So as the pressure of carbon dioxide goes up, the pH of the fluids, of the tissues, will decrease. And so when you have a decrease in pH, you have this bore and root effect. Which again, if this is occurring at the tissue level, if the muscle fibers are contracting, respiring, consuming oxygen, releasing CO2, you will see this effect, which will again induce the hemoglobin at those tissues to release oxygen and in turn absorb that CO2 which is accumulating, which of course is the point. You can see the same effect here with respect to the pressures of carbon dioxide for bullhead hemoglobin, but you can see that the bore and root effects that are observed are substantially less obvious. So the curves are not as different as the previous slide illustrated for trout. And again, this is because bullhead hemoglobin is so much stickier and has a much greater affinity for oxygen. So you can see that the shift to the right is almost indistinguishable, and you can also see that the pressures of CO2 necessary to induce even this minor shift, even these relatively meager bore and root effects, is quite a bit higher. So whereas on the previous slide we saw CO2 accumulation at 0, 3, and 8 millimeters of mercury, here we're seeing 0, 10, and 20, and in spite of that increased abundance of CO2, the magnitude of the shift to the curves is still quite small relative to what we saw for trout. So again, the ability of bullhead to acquire oxygen at the gill is substantially higher because of that affinity of hemoglobin for oxygen, much stickier hemoglobin in this case. But it also results in a substantially more difficult task in terms of delivering that oxygen to the tissues when they need it. And so essentially what this figure is telling you is that the tissues have to endure a much greater degree of CO2 accumulation before any of that hemoglobin will release the oxygen it's carrying at the tissue level. Okay, so this next figure is a little bit complicated here, so we're going to step through it. This is essentially the chemical reactions that are involved in oxygen uptake at the gill, as well as CO2 offloading at the gill, and then ultimately the reverse processes at the tissues. So on this figure, the dividing line is essentially dividing the bloodstream at the gill, which is on the bottom, and the bloodstream at the periphery, which is at the top. Okay. At the center is our red blood cell, so the equations that are shown inside that red oval are reactions that are occurring inside the red blood cell, and any of the reactions that are shown outside of that are occurring in the plasma. Okay, so we're actually going to start with the bottom of the figure, where we have oxygen in the water, so this is dissolved oxygen in the environment, and that is being diffused into the plasma and into the red blood cell as a function of the concentration gradient being in favor of that movement. Again, remember that the countercurrent exchange at the gill is going to facilitate that natural diffusion of oxygen from the environment into the plasma as a result of its concentration gradient. Okay, so oxygen that is in the plasma will ultimately be taken up by the red blood cell and attach itself to the hemoglobin molecule. So again, the hemoglobin is shown here as, it's abbreviated as HB. So we have O2 plus HBH. That HBH is hemoglobin that is bound to a hydrogen ion. So hemoglobin has a greater affinity for oxygen than it has for hydrogen, and so in the presence of oxygen, hemoglobin will release hydrogen and then bind to oxygen instead. And so that's what you see as a function of that equation there. So the arrow moving to the right, you see O2 plus HBH results in HbO2, so hemoglobin bound to oxygen, plus that one free hydrogen ion. Okay, that hydrogen ion can then combine with bicarbonate, that's the HCO3 minus anion there. The bicarbonate anion plus hydrogen ion can be converted to CO2. This is essentially a reversal of the bicarbonate buffering system by means of an enzyme called carbonic anhydrase, and that's abbreviated there as Ca. So as the hydrogen ions accumulate, having been displaced from hemoglobin by oxygen, as those accumulate, that's going to push that chemical equilibrium to utilize bicarbonate ions and then force the greater formation of CO2 through the effect of carbonic anhydrase. As a result of that equilibrium being shifted back to the left, and so we have increasing abundance of CO2, CO2 will accumulate in the red blood cell and in the plasma to the extent that it is at a greater concentration in the plasma and the red blood cell than it is in the environment, and so it will naturally diffuse across the gill and be released. Okay, so chemically, this is essentially the process, the means by which oxygen is taken up at the gill and CO2 is released. It's a matter of pushing chemical equilibria back and forth and establishing and utilizing concentration gradients to facilitate this exchange of these two gases. One thing that I want to point out here is that because oxygen becomes immediately bound to the hemoglobin molecule, that keeps the apparent concentrations or the dissolved levels of oxygen in the plasma very low. Again, once the oxygen is bound to the hemoglobin, it doesn't count in terms of the partial pressures or concentrations of oxygen in the plasma. It's just not in the plasma anymore. So that's going to continue to keep the concentration gradient of oxygen in the water relative to the plasma and the red blood cell in favor of continued diffusion of oxygen into the bloodstream. Okay, so now we've got our red blood cell. It's loaded up with oxygen. It's dropped off its CO2 and its hydrogen ions. And so now it's circulating through to the periphery, and it's ready to find tissues that are in need of oxygen and have to offload their CO2. Okay, so once we get to those tissues, you can see that the levels of CO2 in the tissues, the levels of CO2 in the tissues are going to be quite high. Again, this is because the tissues are going to be respiring anaerobically, so they're going to be generating, the tissues are going to be respiring either aerobically or anaerobically, generating CO2 in the process. And so the CO2 levels are going to be high in the tissues, the O2 levels are going to be low. So what does this result in? Well, the presence of CO2 is going to result in an acidification of the tissues, their fluids, and the plasma. Again, CO2 acts like an acid, so high levels of CO2 are going to drive that chemical equilibrium at the top for the formation of bicarbonate and hydrogen ions. And it's actually that hydrogen ion that you can see there that results in the acidification of the blood. Increasing abundance of hydrogen ions is equivalent to a decreasing pH. So in the presence of that decreasing pH, we have the Bohr-Root and Haldane effects taking place. So the hydrogen ions that are accumulating, they interact with the hemoglobin, which is currently bound to oxygen, and they force the oxygen to become disassociated from the hemoglobin. So you can see a reversal of the equation that we saw before. Now we have hydrogen ions becoming abundant on the right-hand side of the equilibrium. Those added to HbO2, hemoglobin bound to oxygen, result in the dissociation of oxygen from hemoglobin and the binding of hydrogen ions to the hemoglobin molecule. Now, again, as I said before, hemoglobin has a greater affinity for oxygen than it has for hydrogen ions, but the bottom line is that if hydrogen ions become exceptionally abundant, there will eventually be so many of those hydrogen ions that they will force the oxygen molecules to become released from the hemoglobin. So even though the affinity for oxygen is greater, that affinity can be overridden by increasing abundance of both CO2 and hydrogen ions. Okay, so the Bohr and Root effect take place, resulting in dissociation of oxygen from the hemoglobin. That oxygen now does contribute to the partial pressures, or the concentration of oxygen dissolved in the intracellular fluid of the red blood cell and in the plasma, and so the oxygen diffuses down its concentration gradient into the tissues that need it. Now, one other thing that you can see here shown on these diagrams is two transporters shown in green. These are essentially exchange transporters that are utilizing chloride ions to exchange for bicarbonate ions. Many of the processes that we're going to be discussing, especially in the osmoregulation lecture, have to do with the exchange of anions for other anions, cations for other cations, to allow these chemical equilibria to be balanced. So as hydrogen ions are consumed, they are often exchanged for chloride ions to balance charges. And again, that's illustrated here on this figure. Now, one thing I should have noted prior to leaving the previous slide is that that information that's shown on that slide, the process and chemical equilibria involved with hemoglobin and red blood cell oxygenation at the gill and deoxygenation at the tissues, is one that you need to be very familiar with. I can guarantee that there will be a question on the exam that addresses that information. So please be very familiar with those processes of oxygenation and deoxygenation that were shown on the previous slide. Now, this slide is going to illustrate a few pieces of additional morphological information, morphological attributes that can also influence the effectiveness of the respiratory structures in acquiring oxygen and offloading CO2. So you can see a range of different species listed here along the left-hand side, and they're categorized as being sluggish species, active species, or very active species. So in terms of swimming behavior, this tells you whether these are mostly lion weight types of predators, bottom feeders, species that don't actively swim for a majority of their life. The active species certainly swim a lot more. The very active species, like the herrings and mackerels, are continuously swimming. So you can see, as a function of their activity levels and therefore their demand for oxygen, you can see that there are several different attributes here and differences with respect to the morphology of their gill structures. So the first column here is the thickness of the lamellae in micrometers. So you can see the more sluggish species at the top have much thicker lamellae. These are secondary lamellae, much more so than the ones at the bottom, the very active species. This is essentially a function of the behavior of these species and where they live. Thinner lamellae will facilitate more rapid and more efficient gas exchange. It's easier to move molecules through a thinner membrane. But unfortunately, for the species like the ice fish, especially the bullhead, these species, first of all, don't require as much oxygen, so they wouldn't necessarily benefit as much from a more efficient process. But having exceptionally thin, delicate gills would actually be a disadvantage for many of these species, given the environments that they live in. You can imagine a bullhead, for example, mucking around in very turbid water in the benthos. Having very, very thin, delicate gills is going to predispose that animal to gill damage. Very delicate gills will be very vulnerable to abrasions and the effects of turbidity and particulate material that can be damaging those delicate structures. So in the case of those more sluggish species, their trade-off is they have reduced efficiency in terms of absorbing oxygen across their thicker lamellae, but the benefit is that their lamellae are more robust and less vulnerable to physical damage. The next column here is the number of lamellae, secondary lamellae, per millimeter of primary lamellae. So you can see again a pattern here in that the sluggish species have fewer lamellae per unit length of primary lamellae, and that increases as you move into the active and the very active species. So essentially, per unit length of primary lamellae, the very active species have more secondary lamellae within that same space. And again, this is just a matter of increasing surface area. The more surface area you have, the more opportunity you have for gas exchange, which of course corresponds with the greater need for oxygen in those very active species. Of course, if you have more secondary lamellae per unit length of primary lamellae, the distance between the secondary lamellae is going to decrease. So the sluggish species at the top have fewer of those secondary lamellar flaps along each of the primary lamellae, and they're more greatly spaced out. Of course, the species at the bottom of the figure have less space in between each of those lamellar flaps. But again, keep in mind, all things are relative. We're talking about 75 micrometers in this case for the maximum distance between the lamellae, as opposed to 25 micrometers. For all of these species, we're still talking about very, very small distances between the lamellae. They're really packing in a lot of surface area into each of those gill structures. Now, with respect to the distance between the blood and the water, the final column here, this is essentially a measure of the thickness of that single epithelial layer that represents the secondary lamellar membrane. So if we think back to those histological cross-sections of the gill, you saw that each of the secondary lamellae were essentially a flap held apart by the pillar cells, and the flap itself was comprised of that single layer of epithelial cells. Now for the more sluggish species, again, to take advantage of having a little bit more rugged gill structures, their epithelial layer is a little bit thicker. So their single layer of epithelial cells might be 6 to 10 micrometers thick, whereas for the very active and active species, we're talking about very thin epithelial layers, 2, 3 micrometers or perhaps even less. That thin epithelial layer will allow for very rapid and easy exchange of gases at the gill, but of course it makes it a very, very delicate structure. And so it's really only necessary for those species that truly require a tremendous amount of oxygen uptake in a very short period of time. And keep in mind, too, that if we're talking about comparing, for example, a bullhead to a herring or a mackerel, swimming through open ocean, the most turbid particulate matter they may encounter in terms of the environment is zooplankton that are flowing through the water column. As filter feeders, in the case of herring, most of that particulate matter is going to be captured by the gill structure itself. Through mucus nets and that sort of thing. So even that very low level of turbidity is never really going to be affecting the secondary lamellae. It will be captured before then. For the bullhead and these other species that inhabit much more turbid, much more degraded environments, that's certainly not the case. And so having thicker, more rugged lamellar structures is certainly an advantage in those species.